Hi, welcome back to another Poulter's Pod. This time we're going to do a bit more recapping with our organisation topic on an area that I think is particularly important, underpins a lot of this topic, and this is the idea of exchange surfaces. So we want to make sure we've really got these concepts and maybe go a little bit further, but that stretch and challenge in there to really kind of promote your understanding. So what I've done with this lesson to make it a little bit easier for you guys to follow, I've sort of split it into 4 sections. The first section is just a very simple kind of introduction to the problem and why we need exchange surfaces. The second section is probably the longest, and that is where you're really getting to grips with the kind of structural adaptations and the functions of those structural adaptations in our various exchange surfaces. The third section is going to be where you're applying that knowledge to answer a 6-mark question, exam question. And then the fourth section, which is quite important, this is for separate science only, is to look at how exchange surfaces work in fish gills and in insects, their gas exchange system there. Not that we're going to go into epic detail, but we do need to know a little bit about it. So I'll cut to in a second our first key section, some of the ideas behind it, and hopefully they'll make it easier for you to correct the sentence that you need to write. Okay, so section 1. Now, if you're a single-celled organism like a bacterium, life's pretty easy for you in terms of exchange. And when I use this word exchange, I'm referring to diffusion, osmosis, active transport, the movement of substances in and out of cells, which is vital for their function. Every single cell on the planet requires a constant supply of oxygen and glucose so it can carry out respiration. But at the same time, cells produce metabolic waste like carbon dioxide, urea. They have to be transported out. If you're a single-celled organism like a bacterium, that's nice and easy for you because you've got a single cell, you've got a very large surface area to volume ratio, about 6 million to 1. And what I mean by that value is that your surface area is 6 million times higher than your volume. As a result, exchange happens really quickly. The rate of diffusion is very high. You can get that oxygen into the centre of that cell very quickly and carbon dioxide out. But as cells get larger, and if I add more cells to make a multicellular organism like me, your surface area to volume ratio, the relationship between these two variables, decreases significantly. The larger the organism, the smaller its surface to area volume. The smaller its surface area to volume ratio is, and that becomes a big problem. An organism like me, sort of a medium-sized organism, it would take far too long for oxygen to diffuse through my skin to get to the centre of my body. It just wouldn't function. The cells in my interior would die far too quickly. They wouldn't receive those molecules quick enough. So larger organisms, animals and plants, have developed exchange surfaces, structures, to help them overcome this problem, to increase effectively the surface area of parts of their bodies to enable faster rates of diffusion, osmosis, active transport, or a combination of all of them. So that's what I'm going to move on to in the second section, is looking at those, the adaptations that help with that. Okay. So, and I've really hopefully drilled this in, certainly with my set, I've really drilled this into you, that there are 3 key adaptations that all exchange surfaces have. Okay? 1, they've got a very large surface area. 2, they're very thin, 1 cell thick, either way you want to word it. And also, they are often associated with a good blood supply, or in the case of plants which don't have blood, they're close to the xylem or the phloem to maximise things going on. So they're those 3 key adaptations. Hopefully you already remembered those. But they're the kind of structural bits. You need to know why they help. How do those 3 adaptations, which all exchange surfaces have, actually work? What function, what purpose does it serve? So I'm going to go into and talk to you about that now. So in order to get a larger surface area, this could be done through having folding or having many of the structures, and often it's a combination of the 2. So for example, in my lungs, I've got 300 million alveoli. So I've got many alveoli, and each alveolus, which is singular, is folded, and that greatly increases the surface area. But why do I need to do that? Well, if I've got a larger surface area, I'm going to have a higher rate of diffusion. Okay, and therefore oxygen can move from the alveoli into the blood, and carbon dioxide can move out of the blood into the alveoli very quickly, very rapidly. So rapid exchange. being thin, one cell thick, is essential because the longer the distance that a particle has to travel, the slower it will diffuse. So the easiest way of wording this is, exchange surfaces are one cell thick, which gives them a short diffusion distance. The shorter the diffusion distance, the faster it would be. Again, if we go back to the idea of me, and if I was absorbing oxygen through my skin, you've got to think, how many cells are there between my skin and the centre of my body? There's going to be probably hundreds of millions of cells. That's going to take a very, very long time for oxygen to diffuse through all of those cells, whereas my alveoli are literally one cell thick, very, very thin cells at that. So it's a very short, rapid diffusion distance to get into the blood. Capillaries as well, there's an exchange surface as well, often people forget about that. One cell thick with the capillaries, and therefore materials can move between the blood and the cells. Our third factor, our transport system being close to having a good blood supply, one we tend to think about, is a little more difficult to understand, and I think often students do struggle with this. The easiest way I can explain it is I use my model of the idea. Imagine I'm Amazon.com and I've got my factory, I've got all of my packages in there ready to go, and then I've got to send them off to shipping, send them off to be delivered. Now if I've got no delivery drivers, no vans to do that, I'm going to start moving all those boxes outside and they're going to pile up. Eventually I can't get more boxes out the door. I've still got loads of stock and customers to serve, but I can't get it to them. But if I've got a transport system, a delivery system, they're going to take away the boxes as quickly as I can add them. All right. So what that does is it always maintains what we call a steep concentration gradient. So always going to have a big difference between high and low concentration, and therefore diffusion, which is the movement down that concentration gradient, is always going to be very rapid. Okay. There's actually a fourth feature that not all exchange surfaces have, but some do. Lungs is a good example, and also fish gills have this too. And this is something called ventilation. And in the case of the mammalian lung, we are constantly bringing in new air and removing old air. And again, that same process applies. That aids in maintaining that concentration gradient, because of course as I breathe in, my lungs inflate with all that air. The oxygen is going to very quickly diffuse into the blood because that's got a nice steep concentration gradient. But eventually all that oxygen is going to leave. So in order to keep oxygen coming into my blood, I have to ventilate. So I have to exhale that air, bring in a fresh lung full of air, which has got more oxygen, a high concentration again. Where exchange surfaces require active transport to help with exchange, say for example the small intestines or plant roots, where you're absorbing molecules against that gradient, i.e. low to high concentration, then we need to do active transport. And now the cells that make up the lining, the actual surface of that exchange surface, will have various key adaptations. The most obvious one, if I've got to do active transport, I need energy. Well, how does a cell get its energy? Well, by the process of respiration. Where does that happen in the cells? Well, of course, the mitochondria. So when you're wording it in the exam, you would very simply make these kind of key steps. Cell has many mitochondria, so it can then do more respiration and release more energy. As a result, more active transport can take place. Active transport also requires these little structures called carrier proteins in the cell surface membrane, and they're proteins, so having more ribosomes, many ribosomes, will help with manufacturing those carrier proteins, which is all going to help with active transport. Okay, so that's section 2, a little whistle-stop tour there of all the different adaptations and the reasons why we have them. In section 3, I'm not going to say too much, other than, use that information, use the information from the lesson. I've also given you, if you need a little bit of support, some of the key words you could use in your busy bee for that 6 marker. But think about the structural and functional reasons for both why the alveoli and small intestine have them. Okay. So for section 4, we want to take that information that we know about the exchange surface, stuff we know really quite well, and then apply it to maybe some scenarios where it's less obvious to you. And this is also with fish and insects. Now, fish is quite easy to understand. Basically, if you've ever looked at a fresh fish and you've peeled back the flap of the gill called the operculum, you'll actually look inside and see these sort of very, very bright red little, well, they look like hairs, they're actually called filaments. Okay, so if we were to look at this sort of structure here, we would see something like this with lots and 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 lots of filaments. Okay, now already hopefully you're thinking, well, yeah, I know what that means. Is many filaments increases the surface area. Brilliant. Each filament obviously is folded to increase surface area as well, so that really helps. The reason they're bright red, hopefully you've got a bit of an idea with that one as well. They've got an excellent blood supply, again to maintain that steep concentration gradient. All right, they're only one cell thick, short diffusion distance, so everything you've seen before exactly applies here. I've also seen gills for animals like axolotl, which is an amphibian, which has also got these big feathery external gills. And again, no real difference there. You know, it might look a little bit odd to what you've seen there, but you'll see these little feathery extensions on here. Again, many of them increasing the surface area. They're usually quite bright red in colour. Again, good blood supply, one cell thick. So nothing there should really surprise you. When we're looking at insects, it's a little bit more difficult. I'm going to cut to a little diagram in a second that I'm going to draw to give you a little bit of a better idea. Actually, I'm not going to draw it, I'm going to put an image up to help you understand what's going on with gas exchange in insects. Okay, so with gas exchange in insects, it's quite a different sort of structure. The same sort of rules apply though. What insects have got are lots of little holes that run along the side of their bodies. They're called spiracles, okay? And those little holes are a bit like the holes in our face, which allow air to come in, so like our nostrils and our mouth. But instead of having them just localised on their head, they're all the way down the length of their bodies, okay? Those little holes then connect to little tubes that we call tracheae, and they start to travel into the centre of the insect, and then they branch off into finer little tubes called tracheoles. Those little fine branches then actually start to extend all the way around all the different body cells, the body tissues, and then gas exchange takes place directly within the actual body cells. In us, we've got exchange surface, but then we've got our blood, which is our transport system, to move that oxygen throughout our body cells. In the insect, it's direct diffusion within the body cells as a result. So insects do have blood, but it doesn't actually transport any oxygen whatsoever. In fact, their blood is sort of whitish colour, no hemoglobin required, because they've got direct diffusion. So unusual system. I've not really seen any exam questions at GCSE yet on insects, and I doubt they'll ask you too much, but maybe remember those keywords like spiracles and tracheoles. Those of you that are doing this for A-level, and hopefully many of you will indeed look at a lot more detail at insects, and some of what I've taught you now will be totally relevant and helpful. So hopefully that's given you a little bit of an overview of our exchange surface again, and recapped it all for you nicely and helped quite a bit with your knowledge. So this is Dr. P signing off. Have a good weekend. Cheers.